Talking about meiosis and sexual reproduction. Cell division, asexual reproduction. Before we talked about mitosis, where basically we produce cells with basically same information. Basically we produce just identical daughter cells, and these were exact copies, or we can say clones, and they had the same amount of DNA, same amount of chromosomes, and same genetic information. Well, now we're gonna get into asexual reproduction. So organisms that go through asexual reproduction include single-celled eukaryotes and simple multicellular eukaryotes. So yeast and protists are common, basically single-celled eukaryotes that go through asexual reproduction. It includes amobia and paramecium. Hydra, which are some simple multicellular eukaryotes, also go through asexual reproduction. So what are the disadvantages? What are the advantages? The advantage is that asexual reproduction is just actually way faster than sexual reproduction. But the disadvantage is there's no genetic diversity. It's very limited, except for mutations. But it's very limited compared to sexual reproduction. Now, how about the rest of us? Since we're complex multicellular organisms, we reproduce not through asexual reproduction. It's actually through the joining of an egg and sperm. Do we do that? Do we make egg and sperm by mitosis? No, we actually go through this process called meiosis. If we did go through mitosis, we would have 46 chromosomes in the egg, 46 chromosomes in the sperm, and a zygote of 92. But we know that all of us have 46, so that can't be the case. So that's why we actually have 23 chromosomes. So as we can see here, it's a karyotype of a female. We have 23 pairs of chromosomes, and with this female, we have 2 X chromosomes. Now this is a human male. A human male karyotype will also have 23 chromosomes, but our sex chromosome will have an X and a Y. We're going to go through the chromosome some clature naming. So homologous chromosomes are just paired chromosomes that share the same genetic information that they're going to both encoding. They control the same inherited characters and they have the same information. So this is just kind of how we get double-stranded homologous chromosome. So how do we actually make sperm and egg? Like I said before, we must reduce the 46 chromosomes to 23, and this is through meiosis. So basically, the gametes are basically just another name for the sex cells, such as egg and sperm. And when they fertilize, we get a zygote, which is just going to be 46. And that's actually how we produce babies. So now we're going to go more deeper to meiosis. So meiosis is just the alternating stages. Chromosome number will be reduced from diploid to haploid. Diploid is 2N, or 46 chromosomes, and haploid is 23. Meiosis, as we said, reduces chromosome number. It will make gametes. And while, even though the chromosome number is reduced, once we join the two gamete cells and they both fertilize, fertilization just restores that number back to 46. This is kind of just like the cycle. So this is the sexual reproduction life cycle, and this is just an overview. We have our gametes here, with one copy. They're both haploid. When they join together through fertilization, we get 2 copies, and then when we want to start creating maybe sperm reproduction or oogenesis, we'll create gametes, right? So basically through meiosis, we'll just create more gametes like we see here. So meiosis. Meiosis is also known as reduction division, since we're reducing the number of chromosomes from 2N to 1N. The main purpose of meiosis is basically to make gametes. Over here, as we can tell, meiosis actually evolved from mitosis. So it's really similar, but the processes are radically different. So we shouldn't confuse the two. So first, we get interphase 1. Interphase 1 is basically where we just everything, like we discussed from the cell cycle video. We'll just go through G1, S, and G2. Then we have prophase. Prophase 1 is actually a very important cycle in the meiosis, which we'll talk about more later. Then we have metaphase 1, and then we have anaphase 1. All cycles of meiosis 1 are basically the goal is to separate the homologous chromosomes. Then after that we get telophase 1, where we finally get two daughter cells, but they're basically recombinated. And then after that we have prophase 2, metaphase 2, anaphase 2, and telophase 2. And this is a phase that's really similar to mitosis, because we're basically doing the same thing. We're just separating sister chromatids. So important is DNA replication only happens in the first division of meiosis. It's kind of like how mitosis is. It only goes through the first division, so the second division we won't have another DNA replication in between meiosis 1 and meiosis 2. Before meiosis 1, we'll only replicate DNA. So the first division's main purpose is to separate homologous pairs, and the second division's main purpose is to separate sister chromatids. So preparing for meiosis. So prepare my meiosis is kind of similar to how we prepare for mitosis. Basically, we'll just duplicate DNA in the first step of meiosis. And this is, as we can tell, there's a lot of similarities because meiosis actually evolved after mitosis. So we kind of use similar stuff. So in prophase 1, it's very important because this is where the genetic this exchange occurs. So basically, the homologous chromosomes will will be at the synapses, or it's called synapses. Basically, crossing over will occur where homologous chromosomes will form a chiasmata, which is kind of like an X-shaped region, and they'll cross over and swap lengths. And basically, as we can see here, the X forms from in prophase 1. They'll basically just exchange and cross over, which shares genetic information between the two homologous pairs, and that's how we get genetic variation. So this is more about crossing over. It's during prophase 1 where synapsis is formed, or a tetrad forms, and then basically if the region where crossing over occurs is the chiasmata, and basically that's how we get genetic variation. Three main steps is crossover, the breakage of DNA to where we swap, and then refusing the DNA. Meiosis 2 is the second division of meiosis. It'll separate sister chromatids. And as we can see, this is actually very similar to mitosis, except in this case we form 4 daughter cells, not 2, and they're all genetically different. So these are just the overview of the steps of meiosis. As we can see here, in meiosis, basically, we start off with a diploid and we turn haploid. Meiosis 2, we have a haploid, and in the end, we'll just have another haploid. These are some informations of meiosis 1 and 2 and some graphic and visual display. As you can see here, like metaphase 2, for example, we can see the sister chromosomes is kind of lined at the center, getting ready to split. And anaphase 2, we can see them on opposite sides of the cell. This is an overview of mitosis and meiosis. Basically, the main difference is basically just what we discussed about how meiosis 1 is homologous, basically splitting apart, and meiosis 2 is sister chromatids splitting apart. This is important, maybe a chart you want to like take notes and identify. These are just some main differences between them. How mitosis produces somatic, while meiosis produces gamete. And mitosis has no crossing over since we have identical cells, but meiosis has crossing over, which produces genetically different cells. And in the end, through this cycle, well, after fertilization, we'll form a zygote. Now, what is the value of sexual reproduction? As we can see, sexual reproduction introduces genetic variation, which is very important, especially in the time of where uh, diseases and everything. Like, if all of us had the same like genetic variation, none of us would be able to evolve and adapt to the new surroundings that the Earth keeps changing. We something we'd easily, for example, be wiped off the Earth. For example, if any disease or anything came and everyone shared the same genetic variation and no one adapted or no one could evolve to have basically thrive in specific conditions, we'll all just be wiped out. So genetic recombination occurs in like through independent assortment, which is just the random assortment of chromosomes crossing over like we discussed, and random fertilization, like which sperm fertilizes which egg. There's so much sperm that's going to fertilize an egg. So through random assortment, we actually have 2 to the power 23 combinations, and that just shows like how genetically different we are. From crossing over, we'll actually create an infinite variety of gametes. And in random fertilization, any two parents will provide a zygote. So that's 2 to the power 23 from independent assortment to the 2 to the power 23. And if we multiply those, we get over 70 trillion. So that kind of makes sense how none of us are actually look the same, or none of us have the same genetic information. So sexual reproduction allows us to maintain both genetic similarities and differences. So as we can see, that's why you might look like your brother, for example. As we can see, like we with brothers, like the Jonas brothers, they have a genetic similarity because in the end, all their DNA comes from their parents that's shared. But that doesn't mean they're going to get the same proportions of genetic information from not proportion, but the same. For example, genes from your father or your mother. This is just sperm production, which you don't really need to know for AP Bio. Egg production is through oogenesis. This also you don't need to know through AP Bio, but it's maybe good to understand if you really need to. So differences across kingdoms. Not all organisms will actually use haploid diploid stages the same way. Plants will use alternation of generations actually, which is a bit different. Where they have like a dominant phase and then like a sporophyte phase and a gametophyte phase, which is a bit different. And but they still will alternate between haploid and diploid. And that is about meiosis. Thank you for watching.